Hi everyone and welcome to today's Biology Daily Booster. This is number 15 in our Paper 2 series and it is on that advanced information list for both foundation biology and higher tier biology. What we're going to do then is have a look at plant defences. In terms of our plants, they've got two types of defences then, physical ones and chemical ones. Your physical defences in a plant are going to be these physical barriers to stop the microorganisms getting into the plant in the first place. Chemical defences are, as the name suggests, chemicals that will be secreted by the plant to try to kill off these microorganisms. What I've given you at the bottom there is just a little picture of a little cross section through a leaf, just as a reminder of some of those key features. So we've got this top little line here, which is the waxy cuticle. We then come to the epidermis. Then we've got our palisade cells, the ones that carry out photosynthesis. We then come into our spongy mesophyll layer, which has got the air spaces here. And then at the bottom, we've got our guard cells. So there's a guard cell. And of course, our stomata between our two guard cells. There we go. Let's look at the physical defences first of all then. So our first physical defence is the waxy cuticle. So what we actually have covering the epidermal cells are these waxy substances. And one of the key purposes behind that is to prevent water loss from the plant, so that reduces transpiration of course. But it also prevents the pathogens coming into direct contact with those epidermal cells. So what we're doing is we're putting this wax barrier between the cells they can infect and the pathogens themselves. So that reduces the chance of infection occurring. The second thing that we're getting as a defense here is because wax is actually hydrophobic, so hydro, water, phobic, not liking, then the water just runs off the leaf. And you've probably seen this on plants. If you look at them after rain, the droplets tend to just run off quite easily. And that's a big bonus because if we don't have sitting water on our leaves, there's nowhere for the fungal spores to sit and germinate on our leaves either. So it helps protect against fungal infections. Second physical defence is the cell wall. Now, cell walls are a major defence against both the fungal and bacterial pathogens because it's this really sound structural barrier. No matter what plant we're thinking of, they all have a primary cell wall. Now your primary cell wall is made of those cellulose fibres, giving it the strength and the flexibility and gives the plant that structural support. What we find is those cellulose cell fibres are actually cross-linked with other substances, like pectin, and that's going to form basically a cement to join all these cells together really strongly. In a number of our plants, we also find cells that produce a secondary cell wall, which develops inside the primary one. So if you've got the secondary cell wall as well, then it makes it even less likely that any of those pathogens are going to be able to enter the cell. We then come on to our chemical defences. So chemical defences are where we're producing chemicals in our plant to protect against some form of infection. So what we've got, insect repellents first of all, and you've probably used some of these yourself at some point. Citronella is the stuff you get in the candles in your garden or those little bands they sell on flights when you go on holiday to places. And pine resin. Both do the same thing, they just act as a repellent for the insects. They don't want to come near that plant. Insecticides, side means kill, thing in front tells us what, insects in this case. And our example there are pyrethrins. Some plants make antibacterial compounds that work in different ways. So phenols disrupt the bacterial cell wall, defensins disrupt the cell membrane. So it affects obviously that bacterial cell and how it's actually going to survive. We have antifungal compounds. These are going to affect obviously fungi. So chitinases break down the chitin, which is what makes the fungal cell wall. And caffeine is just toxic to fungus. And finally, some plants make cyanide, which is just toxic to any and every living thing available. When it comes to identifying a disease in a plant, then we've got a couple of techniques that we can use out in the field to actually try to work out what disease is affecting our plant. We can carry out observation, where we're just going to look at the visual symptoms. So what's happening to the leaves? Are they discoloured? Are they curling? Is there a deposit on them? Is there some evidence of looking a bit rotten? Is there fruits that are rotting? Are there strange deposits somewhere? So we're looking at visual symptoms there in the observation. Alternatively, we could use microscopy. Remember, light microscopes, they're light, they're portable, we can take them out into the field. It's dead easy to prepare a sample for it. So that means that we can have a little look at the particular bits under the microscopes and see what type of pathogen it is. Usually light microscopes will work for this, but there are some that we need to use an electron microscope for. And obviously, as we said in our first paper, that's not feasible in the field. So that would have to obviously be sent to a lab for electron microscopy to be carried out. One thing to note though, when we're talking about working out the kind of pathogen that's affecting our plants, if we're talking about our field observations, these are going to only be possible once an infection has taken hold. It's not that you're just going to wander through a field and think, yep, that plant's going to get this disease. Unless we can see the symptoms, so the disease is already in full swing, we won't be able to do it through these methods. Which brings us on to our second potential technique, the ones in the lab. So we can use DNA analysis, which is basically where we're going to run our little DNA samples through an electrophoresis gel. It splits the DNA into little bands, and then we can compare our unknown pathogen to ones we already know. And all we do to identify it is match the bands. So in this case, we can see all three of them have got that top line, they've all got the second line, but this third line is only present in the one to the right. And you just keep going down, and what we can see is because all of those lines match up for this, we have cucumber mosaic virus as the pathogen. We can also use the process of antigen identification, and the antigens, if you remember, are those protein spikes on the surface of the microorganism. They are specific to the pathogen, and therefore we can identify them by chemical analysis. So we've got a couple of ways that we can identify exactly what is affecting our plants in the lab: DNA analysis, antigen identification. If there are topics that we've gone through in our booster today that you'd like to go into in a bit more detail, we do have obviously the videos on the B6 playlist on the channel that you can use. Hopefully you've got revision guides, classwork notes that you can refer back to, and it's always worth having a little flick through some of those past exam papers just to get a feel for the style of question that they ask you on this paper too. And of course, don't forget to join us again tomorrow for our next Daily Booster.